Hi, everybody. Welcome to Little Lectures, part of the series run by University of Aberdeen. I am Dr. Ashish Malik, and I'm a lecturer in biogeochemistry at the School of Biological Sciences. And today I'm going to talk about understanding how soil microbiomes can help sequester carbon to mitigate climate change. Much of this work was done at the Center for Ecology and Hydrology and the University of California, where I was doing postdoctoral research projects before I joined the University of Aberdeen in November 2019. So I'd like to thank the great labs I was part of, and this work was made possible by funding from the European Commission, from the US Department of Energy, the NERC Center for the NERC Environmental Research Council, and the University of Aberdeen driving my current research portfolio. Much of the research that I'll be presenting has been published or are available online as preprints, and you can find all of that information on my website. Most of the papers are also open access, so easily accessible. Let's just kickstart with the seminar. We know that soils are very important in storing carbon. The amount of carbon stored in soils is more than the amount in the entire vegetation and the atmosphere put together. And soil microorganisms are very important in the below ground carbon cycling. They not only sequester, they not only, sorry, I can't see my, yeah, that's fine. So they not only sequester carbon, but they also are important in releasing the carbon back to the atmosphere. And plants fix carbon, and this is our high school biology. Plants fix carbon, and the organic matter from plants is then available to microorganisms, and microorganisms then break it down to release it back to the atmosphere as CO2. But new research suggests that microorganisms can also contribute to formation of soil organic matter through remains of microbial biomass, which we often call as necromass. And this is a significant finding which changes the paradigm a little. And we know that fossil fuels emissions because of human activities is releasing a lot of carbon dioxide into the atmosphere, and this is changing the balance of the carbon cycle. And because of global warming and climate change, there are a lot of feedbacks that is disturbing the balance of the carbon cycle. But because microbes are so important in driving below ground soil carbon cycling, they can also be part of some of the solutions to key challenges that exist. And one of them is intensification of land use, which is causing loss of carbon from soils. Some estimates suggest that because of agricultural intensification, there is the loss of carbon is around 133 gigatons of carbon, historically speaking, which is equivalent to the amount of carbon that's lost from the vegetation due to deforestation. And if we compare that to the amount of carbon that exists in the current pools, that's quite significant. It's about 10% of the carbon that exists in the soil, which has been lost because of intensification of agriculture. So if we want to keep this carbon in the soil, we need to reverse this trend of intensification and move towards sustainable agriculture so that we can keep that carbon in the soil, and we can even enhance carbon sequestration in currently degraded soils, which can help us mitigate climate change by keeping the carbon in the ground. And I'm going to try and convince you through this talk that microorganisms in soil are important in helping us achieve this goal. So we know now that soil microbes are gatekeepers of soil carbon atmosphere exchange. They can change the balance because of key processes they play an important role in, and that's decomposition and stabilization of carbon in soils. So once microbes have access to plant organic matter, they use it for various purposes. So the box that you see here is the microbial biomass in soils. So all the microbial communities that exist in the soil. Now, individual microbes in the soil will respond to their environment and choose to invest in different processes, different metabolic pathways based on the resources they have. And depending on whether they invest in growth yield or they invest, whether they invest in biosynthesis or whether they invest into respiration, which fuels a lot of the maintenance processes, that will determine how much of that carbon remains in the soil and how much is lost back to the atmosphere. So for example, if there are limited resources in the environment, then microbes will have to invest a lot of their carbon and energy into resource acquisition strategies in the form of extracellular enzymes or transporters, in the form of resource discovery and motility to reach the resources. So this investment in resource acquisition can have a trade-off with growth yield, in the sense that they will not be able to grow as well as they would in a resource-rich environment. Similarly, soils are full of extremes of abiotic conditions, which act as stress factors on microorganisms. So depending on the kind of stress in the environment, microbes will also have to invest their carbon and energy into stress response functions, stress response metabolic pathways, and this can come in various forms. For example, if the soil dries, they might have to invest in maintaining osmolarity because the outside osmotic pressure, osmotic potential decreases. So they have to secrete special compounds like osmolytes, or they have to secrete extra polysaccharides to act as sponges. And this investment in stress response can again have a trade-off with growth yield. So they invest less into biosynthesis. And why is the biosynthesis investment so important? Because ultimately, when microbes die, the dead biomass or the necromass then contributes to stabilization of carbon in the soil as soil organic matter. And this happens when the microbial compounds interact with the mineral matrix in the soil, and this organo-mineral interactions are then strong, and this is how carbon is stabilized in soil. So to really look at how carbon sequestration can be enhanced, we need to look at the microbial physiology. And we've come up with a trait-based framework which tries to look at these three main life history strategies of microbes: of high growth yield, resource acquisition, and stress response. We call it YAS. And based on how microbial species are structured in the environment based on gradients of resources or stress, their life history strategies would change. And this structuring along these three life history strategies will determine how carbon is stored or lost in the soil. So I'll give you an example of how we tried to look at this trait trade-off strategies and how microbial physiology can impact soil carbon using samples from all over the UK. We had 28 sites, and each of the site had a local land use contrast. So we wanted to look at the impact of land use intensification on microbial physiology and the consequence of that response of microbes in soil carbon sequestration. So we have these local land use contrasts next to each other of more pristine grassland versus high intensity agricultural cropland. And this is not a perfect experiment because we chose to work with farmers and landowners who had some of these contrasts available. So the treatment is not the same, but it's always comparing a more pristine site with a more intensive agricultural cropland. And what this allowed us is to minimize the impact of climate and parent material, the soil properties on the microbes, but only looking at the impact of land use intensification. So what we did was we took soil cores from each of these sites in triplicates, and we measured the community carbon use efficiency, which is shown here on the y-axis. And this was measured, this is a proxy of microbial growth yield. So higher the carbon use efficiency, higher the amount of carbon that is invested into growth. And we measured this by using an isotope approach, which we use a lot in my research group. We used 13C labeled plant litter dissolved organic carbon. So this is a solution that's made from 13C labeled plant litter that was powdered. And we looked at how much of that 13C, when you give it to the soil microorganisms, gets partitioned between the microbial DNA and the respired CO2. And this can then give us an indication of how much of that carbon gets transformed into biosynthesis, growth, and how much of that is lost back to the atmosphere as CO2. And on the x-axis here is the carbon enzyme activity per unit biomass. And this gives us the enzyme investment by the soil microbial communities. And this was measured using fluorometric enzyme assays. And we used two enzymes to capture the carbon enzyme potential per unit biomass. And a higher investment in extracellular enzyme would mean higher investment into resource acquisition strategies. So that's shown here on the x-axis. And so what we found was a negative relationship. So higher the enzyme potential per unit biomass, lower was the community carbon use efficiency or the community growth yield. So for example, in soils where the investment was much higher into extracellular enzyme production, they were less able to invest into growth and biosynthesis. And if we overlay the amount of soil carbon in each of these soil cores, we see here that the lower carbon concentrations were mostly in the intensive agricultural cropland soils. And these soils are here on the right side at the bottom, where you can see that they have high investment in extracellular enzyme production because the organic matter here is less in the soil, so they have to mine for resources using these extracellular enzymes, and this takes a toll on growth. So they are not able to invest as much into biosynthesis and growth, whereas in the pristine habitats, which had higher soil organic carbon, resources were ample, so they didn't have to invest into the extracellular enzymes, and that means that there is no trade-off with other investments like growth, and they can really have high growth and turnover and have high carbon use efficiency. And we see that that then leads to higher carbon sequestration as well. So we then also looked at soil proteins, and we do this a lot. We use these modern omics technologies to look at the functional potential of microorganisms. And with these tools, you can look at the community level functions in terms of which functional genes they have, which functional proteins they have, but you can also look at the taxonomy, who are producing that. And this is a tool that is transforming microbial ecology. What we did here was, again, look at some of the sites, and we did soil proteomic analyses. And I'm showing here the pairwise indicators that were unique or significantly higher in each of the two land use treatments. So low intensity are the pristine soils and high intensity are the agricultural croplands. And what you can see here is both in terms of the diversity of proteins relating to ABC transporters that you see, as well as the abundance of those ABC transporters. They are high in the low intensity pristine soils. And this again highlights results from the previous slide where I showed that these soils, the pristine soils with high soil organic matter, had higher resources available. So also in terms of root exudates and available organic matter for microorganisms, these were available to them. And so they just had to produce these transporters to suck in some of these nice juicy substances which are not polymeric, and take them up and use as resources. Whereas in the intensive high intensity agricultural systems, resources were scarce because we are taking away all the plant matter for production of food and fiber. And in these systems, we saw a lot of stress proteins in the microbial communities. And this is an indicator that these systems are also stressed. They are resource limited, but they are also stressed because lack of organic matter also means that physically the soils have a poorer texture, poorer structure. So they are able to hold water less well. So these systems are also drier. And so it's also a bit of a stress for microorganisms. So they have to invest, in addition to investing into extracellular enzymes for resource acquisition, they also have to invest into stress tolerance traits. And these are through stress proteins, which are mostly to maintain the integrity of the biomolecules. So in terms of the microbial community and their physiology, what we found was that in the pristine systems, because resources are ample as available organic matter and stress is minimum, the microbes there can really grow well, reproduce, and they have a high growth yield. And in the agricultural high intensity systems, these soils are poor in plant organic matter because we are taking it away. And they are also drier and have a poorer soil structure. So they have the microbes in these soils have life history strategies that are more inclined towards stress tolerance and resource acquisition strategies. So this means that they are less able to grow well. So in terms of a policy perspective on this front, the science highlights the importance of having restoration or regenerative agriculture, which makes the microbes happier in these soils. So giving them more resources and decreasing the stress will increase the amount of carbon they invest into microbial biomass, and that then can allow us to have more carbon sequestration through that biomass and necromass stabilizing with the soil mineral matrix. So one way of improving soil structure as well as soil organic matter, which is important to sequester carbon, but also to improve soil health, which which likely leads to higher plant productivity, is to look after the soils, look after the soil communities so that they get the resources and they are not stressed, and they can invest more into high growth yield strategies, which can then cause reiterative soil organic matter formation through the necromass stabilizing on the soil mineral matrix. So that is the kind of research we are doing here at the University of Aberdeen in the soil, in the microbial biogeochemistry lab. We are looking at individual species at the population level, looking at how these trade-offs in different traits structure microbial communities. We are looking at the community structure as a whole because that allows us to look at how the physiology is affecting at a scale which translates into changes in carbon pools and fluxes, which can then be linked to ecosystem level functions in terms of carbon sequestration. And these can then help us to improve our projections for the future, or to come up with solutions to some of the challenges like climate change resilience. Some of the projects that we are looking at are trying to study the eco-evolutionary responses of microbial populations in response to climate change induced extreme events such as droughts. We are trying to see how microbes are adapting to a drier environment and what is the consequence of that adaptation on soil carbon cycling. We are also looking at peatland restoration projects because this is a national priority here in Scotland. One of our projects is looking at how the microbes are changing their physiology when degraded peatlands are restored. And this can then help us to look how carbon can be sequestered in these peat systems using microbes as a tool, because we know that when peat are degraded because of water table drawdown, microbes become active and they break down the peat organic matter. So one way of restoring peatlands is to change the conditions for microorganisms so they don't break down the organic matter as fast as they would. So these are some of the key challenges that we are addressing using fundamental microbial ecology coupled to ecosystem ecology, using tools ranging from stable isotopes to ecological omics. And if you need more information on this work, or if you have more questions, please contact us, and we are happy for collaborations, and we are happy to have more students join us. With that, I'm happy to take questions from you. All right. Let me look at the questions that are coming up here. Okay, so the first question is, in what form is carbon stored in the soil? Is it active, inactive, or a form that plants can use, or a form where it can gradually escape back into the atmosphere? Is it very different between natural ecosystems, example forests, grasslands, and agro-ecosystems? Is there a difference between temperate and tropical sequestration? All right, so there's a lot there. So carbon is stored in the soils, like I said, mostly in the form of organomineral complexes. So yes, a lot of plant matter, which is resistant or recalcitrant to degradation, can stay in the soil for months to years, but most of the carbon that's preserved in the soil for decades or centuries is actually the microbial necromass, the dead microbial biomass that's associating with the mineral matrix, and this association is quite strong, and it makes that carbon then inaccessible further microbial excess and decomposition. And this is the carbon that's stored in the soil as soil organic matter. And yes, it can gradually escape to the atmosphere when microbes can have access to this carbon. So the physical access to organic matter is a very important rate limiting step in determining the loss of carbon from soils. About the question on it varying across ecosystems, yes, it varies. In forests, for example, there is a lot of litter that remains on the top organic layer because the above ground carbon is quite high. Whereas in grassland soils, the below ground inputs tend to be more important, and therefore the microbe mineral interactions, higher amount of carbon stored in the mineral soil is likely to be more important in carbon, likely a more important carbon pool in grassland ecosystems. Again, in terms of the differences between sequestration in temperate and tropical climates, again, it depends a bit on the rate of turnover of carbon. In tropical systems, the turnover tends to be high, which is why there is less amount of carbon that can be sequestered along the depths of the soil, whereas in temperate systems, because of colder environments and perhaps also in a lot of environments, high water content, carbon can be sequestered for a much longer period of time. For example, most of the peatlands that we see in the high latitudes store a huge amount of carbon. I hope that answers your questions there. Okay, the next question is how is it possible, considering time and cost, to engineer a number of such microbes to help sequester high volumes of carbons in different carbon content disposed media? So in terms of time and cost, I think this is kind of veering into the geoengineering scenario, and both in terms of time and cost, this wouldn't be very helpful in terms of designing microbial communities that can sequester carbon. Because as I mentioned, our results suggest that if you give the microbes the right conditions, they are able to grow well. And so, for example, in degraded agricultural systems, sometimes the amount of carbon is so low that there is so much potential to sequester carbon in these systems. So if you just give the microbes a little bit of plant organic matter as inputs, it will start to sequester carbon over a period of time when soil microbes start to experience less stress and less resource limitation, and they start investing a lot of the carbon that's being given to them to form biomass, which then stabilizes in the mineral matrix to form soil organic matter. So that, according to me, is one of the best solutions we can do, and this is a pretty low-hanging fruit. Also would like to mention that soils are a slow in, fast out system. So for the plant and other organic matter to get stabilized, it's a very slow process, but the loss of carbon can be really fast, which is another reason why we need to maintain higher levels of carbon sequestration through these microbial processes. All right, there is another question here. how quickly can we expect soils to recover from these intensive farming practices if stress factors are reduced and the appropriate resources provided? So this is not something I've worked on directly, but some of the systems we've worked on, so some of these local paired contrasts, we also have a restoration treatment there. So some of these soils had historically been under an agricultural land use, and then they were left unattended, so that grasslands can rebuild there, and we have chronosequences. So those that were restored for decades, those that were restored more recently, and we see that it takes about 5 to 10 years for the soils to slowly start building carbon. So my guess would be that it takes at least 5 to 10 years, perhaps longer. But this again depends a lot on the kind of soil, the environment we are in, and all of that, because the mineral material is also very important. And if the soils are very degraded, then it's obviously has a much higher potential of carbon sequestration. Okay, there is another question about pollutants. Do pollutants present in the soil affect both negatively and positively carbon sequestration? This is not my expertise, definitely, but I think what it suggests is that pollutants are also sort of stress compounds for microbes, and it definitely affects microbial diversity in some ways. How it affects microbial physiology would be a tough guess for me to make, but I would definitely think it affects my carbon sequestration negatively, because it sort of disturbs the balance in the ecosystem. All right. I'm going to just wait for maybe half a minute to see if there's another question. And if not, we can start to wrap up the session. All right, there is another question which I have just seen, and that's about knowing that carbon exists in several chemical states in different environmental media, soil and air. Which state will the microbes best sequester carbon? So yes, the carbon in the atmosphere is best transformed into organic matter by plants by the process of photosynthesis, but some microbes can also fix atmospheric carbon in the form of carbon dioxide. And so these autotrophic microorganisms can also sequester carbon, but the amount will definitely be lower. And there are some estimates which are suggesting that there is a significant amount of carbon that can be sequestered by microbes by transforming the CO2 into organic matter. But mostly what we know is that carbon in the soils, either from plant organic matter or from other dead microbial organic matter, is the most important source of carbon for microorganisms, at least the heterotrophic microorganisms in soil, and they then transform that organic matter. They use it up, release it back to the atmosphere through mineralization, and also convert some of that into biomass, which can then be sequestered. So I guess those would be some of the ways by which carbon that exists in different chemical states, in different environmental media, can be transformed to form soil organic matter. All right, so if there is no more question, I would like to wrap this now. Thank you very much for attending the session. And if you're listening to a recorded video later on, thanks for watching. Bye for now.